My name is Marlene Romero, and this is my submission for the DNA radio format second. I will be answering the questions that were provided and required for this assignment, and along the way, I will be mentioning the terms DNA, gene expression, and proteins. I will be explaining what is DNA and why is it important, and I hope you guys enjoy it. So first off, what is DNA and why is it important? So DNA is defined as the molecule in cells that contain the genetic instructions for an organism development and function. So DNA is basically the genes from our parents that were passed down to us. It's important because it helps us do our daily routine. Every function that we can do, we're able to do that because of our genes. And it's important as well as because it makes proteins that we need to do our daily task. But it is passed on along the way as we as the population grows, like our offspring will be someone else's passed down DNA. So I will be answering the questions provided in the submission. So first question. First, how is it that different cells in the same organism with the exact same DNA make different proteins from each other? The reason for this is because of two processes called gene expression and what's the other one? Selective expression. So first off, what is gene expression? Gene expression is the process of converting the genetic information in a gene into a functional protein, which is called the gene expression. Meanwhile, selective expression slash process is selective by using different genes to activate or deactivate it. So they basically they do different routines, like processes, but they still get the same DNA. Second question, how do cells make sure that there's enough mitosis but only the normal amount of DNA in meiosis? Cells ensure enough DNA for mitosis by replicating the chromosomes once and then separating them the identical copies into two daughter cells. In meiosis, cells that replicate their DNA once that they undergo two rounds of division. The first one would be the homologous paired chromosome, and then the second one would be the sister chromatids. This results in 4 cells with half of the normal amount of DNA. Third question, final question. Why do you think DNA needs to be copied into mRNA to make proteins rather than directly making protein from DNA? I think for this reason is because DNA is copied to mRNA to protect the DNA, which allows multiple proteins to be made from one gene at a time and separate the processes of DNA replication and protein synthesis. Thank you guys so much for staying through to this video. I hope you enjoyed and I hope you learned some new terms of DNA, proteins, mRNA, and all the above. Thank you.